Okay. All right. Thanks a lot for that introduction. It's great to be here. The subject of my talk will be how basic sensory stimuli are processed in kind of their probability context. The primary method I use is 2-photon calcium imaging, but I also do a lot of local field potentials, and it's all on awake mice. Basic cortical processing of sensory stimuli is significantly influenced by the context in which that stimulus appears. So if this apple were to suddenly appear in a context with a bunch of other fruits, you might expect one level of response in primary visual cortex. Whereas if that same stimulus were to appear in a context where there's many apples, it's expected, it's redundant. What we actually observe is a suppression of responses to that stimulus. Conversely, if that stimulus were to appear in a context where the observer expects oranges, you get a sort of amplified response to that stimulus. So this basic sort of contextual modulation of basic sensory processing has been studied traditionally using oddball paradigms. So what this paradigm involves is the repetitive presentation of the same stimulus over and over and over to the point where that stimulus becomes redundant, and then every now and then throwing in an unexpected or a deviant stimulus. This has been recorded in humans, non-human primates, rodents, and so forth. And the idea, using, for instance, EEG, is that the deviant or the unexpected stimulus elicits a stronger response than the standard or the redundant stimulus. And one way this has been quantified is with mismatch negativity. So what you do is, while recording your EEG response from the scalp, you average the responses to the repetitive stimulus, and you compare that to the waveform of the average response to the deviant or unexpected stimulus. And what you get is this sort of negative going potential here at about 100 milliseconds or so after the onset of the stimulus. So why this is particularly interesting is that we know that individuals with schizophrenia show a smaller mismatch negativity, meaning that they have a smaller difference in their evoked response to the unexpected stimulus versus the expected, or the deviant versus the redundant. And this is relatively selective to persons with schizophrenia versus bipolar disorder or other major psychotic diseases. So, so what this in particular buys us is that if our overall goal is to understand the neurobiology of a complex disease like schizophrenia, what we essentially need is to understand how variation at the genetic level gives rise to sort of changes or pathology at the molecular, cellular, circuit, systems, and eventually behavioral level. We need to sort of understand how these different levels interact, and we need to be able to sort of pinpoint where exactly things kind of converge in this disorder. But one major complication is that in patients, we can't directly access these levels of function non-invasively. So we need animal models so far. And a bigger problem is that how do you study a complex disease like schizophrenia in a mouse? The idea that you could directly observe the presence of delusions or hallucinations or disorganized speech and behavior it's somewhat insulting to our sensibilities in the sense that how do you know if the mouse is psychotic? How do you know if it has schizophrenia? And one way to get around this is by studying context processing. So instead of trying to study the holistic disease of schizophrenia, you study sort of the mismatch negativity, or a biomarker of the disease, something which we know is relatively selectively affected in schizophrenia, but it's perhaps more proximal to the cell and circuit level dysfunction, which might underlie the disease. So this is the general strategy of my work, and this is the way my talk will be set up. First, I'll kind of discuss the paradigm and how I study sort of sensory context processing in mice. And then I'll, after I have that kind of model hammered down, I'll show how this context processing is carried out at the single neuron level, at the level of interneurons, at the level of local populations, interlaminar circuits, granular, supragranular, and infragranular, and then also how this is carried out across distal brain regions. And then looking forward, I'll bring up some sort of ideas and hypotheses as to how these different levels of function interact to give rise to sort of basic contextual modulation of sensory processing. So to begin with, can we study context processing in mice, or does the mismatch negativity biomarker translate well? This will be the majority of the data that I show today will be in this very basic paradigm. We use awake mice, head fixed on a treadmill, and we're showing them full field visual square wave grading stimuli. So in this case, you just present the mouse with the same oriented stimulus over and over and over to the point where it becomes redundant, and then every now and then change the orientation and sort of quantify the responses to the deviant stimulus versus the redundant stimulus. And in order to sort of measure cortical responses, we use 2-photon calcium imaging. This strategy, you virally express GCamp. All the data today are from using GCamp 6S and 30 hertz imaging. So you express these calcium indicators in primary visual cortex. And then this is just data from an awake mouse without the presence of sensory stimuli. You can sort of see here when an individual neuron fires an action potential or bursts of action potentials, the neuron gets brighter in the field of view. So you can get activity and information from individual neurons and also get an idea of how they relate to one another in a local area. So it's a potentially powerful technique. And here's some real-time data. On the left here is the visual stimulus that the mouse is seeing. And on the right is calcium imaging from primary visual cortex during this paradigm. And you can see that the majority of context is relatively silent, even though you have this very big, large, full field oriented grading. But then when the deviant stimulus appears, you get a very large response from a large number of neurons. So if you're having a hard time seeing it, you can just sort of focus on a few of the neurons in the field of view. And then they're mostly silent, and you get this deviant stimulus, and they all come online. So I can let that go through another time or two. But the idea is that this response is robust enough, and it has a high enough signal-to-noise ratio that we can observe it in single neurons on single trials. This is averaging over trials. Each individual row is a single neuron. And I'll show a lot of plots like this. So time is the x-axis. And you can see that the average response to the stimulus when it's contextually deviant is significantly lower than the average responses to the stimulus when it's contextually deviant versus redundant. And the color represents how large the calcium transients were. So this is sort of an index of bursting. So if we come back to this, you can imagine at least two things are going on here. First, you have a large suppression of activity to a repetitive stimulus. So this stimulus is occurring over and over and over to the point where it's redundant, and most of the neurons are not firing at all. Even given what you would expect, basic sort of orientation selective responses in primary visual cortex, you still see essentially nobody firing. But then on the converse, so you have suppression of responses to a repetitive stimulus, but you also have this large augmentation of responses to a deviant stimulus. So this is kind of bidirectional. You get sort of suppression, you also get a deviance detection. And the way to tease those out is by using a number of different paradigms. So first you just run your typical oddball paradigm, where this particular 135-degree grading is your contextual deviant. And then you can run it again by flipping the deviant and redundant. And so you can get responses in the same cells to the same stimulus when it's in a different context, when it's expected. And then you can run a third paradigm, which I'll call the many standards frequency control. And in this case, the stimulus is neither redundant nor deviant. It's in the context where there's many different orientations occurring. So the stimulus is still rare, but there's nothing unexpected about seeing this stimulus. So you have these three different contexts here. Another way to visualize this is just the same sort of schematic, but plotted in a different way. Here in the context neutral control, there's many different orientations, whereas in this particular paradigm, that stimulus is the contextual deviant, and then in this one, the same one is the redundant. So we run each one of these. Each one takes about 15 minutes with about 5 minutes in between. And so here are responses of the, yeah?
Can I just ask a clarifying question? When you're doing your imaging, do you expect most of the cells that you're imaging are layer 2, 3, or do you expect some of them are layer 4? Maybe you said that and I missed it.
Yeah, okay, so this full first half, it'll just be layer 2, 3 neurons. So yeah, it's a good clarification. And a lot of these properties are most dramatic in layer 2, 3. It's kind of a teaser. But yeah, so this is maybe about 150 to 250 microns from the surface. Right. So So here you have responses of the same neurons. So each row is a neuron in layer 2-3 of visual cortex to the same stimulus when it's kind of context neutral. It's neither redundant nor deviant. When it's contextually redundant, and when it's contextually deviant. So if you average over these neurons in each one of these contexts, what you see is this bidirectional effect I was talking about. The black line is sort of your neutral context, and this is the y-axis is sort of z-scored calcium transients. So you can see this sort of one level of response that is expected in this sort of baseline condition. When the stimulus is contextually redundant or repetitive, you see a suppression of responses. And when the stimulus is contextually deviant, you see an amplification of responses. So this bar graph is the same data, just kind of collapsed over time. This suppression of activity to a repetitive stimulus has been referred to as stimulus-specific adaptation, whereas the amplification of activity is what I'll call deviance detection. All right. So this is what's going on in individual neurons when you average over neurons, when you average over trials. I'm also typically trying to record local field potentials during this paradigm as well. So this is from a 16-channel multi-electrode shank from NeuroNexus. And if we just take one of the traces and average over these three contexts, you can see that the sort of average LFP waveform are also modulated by deviant versus redundant contexts. And if we look at what the human EEG response looks like in this case, this is just taken from a scalp electrode. And at least sort of ostensibly, the sort of waveforms look relatively similar when we pick from deeper layers. And you also have this sort of negativity that we observe in mouse visual cortex, which is very relatively similar to what we observe in the human patients. So we can at least say, to some degree, we're recording something that looks like this sensory biomarker of schizophrenia in a mouse model. But what's not typically done in the human literature is to split it up into adaptation versus deviance detection. And what you can see from LFP, which has a little bit better time resolution, the sort of adaptation response is occurring a little bit earlier. So the blue line is the average response when the stimulus is redundant. And sort of that sort of stimulus-specific adaptation occurs about 100 milliseconds in, whereas your deviance detection, or your major difference that's elicited by unexpected stimuli, occurs a little bit later. So we also have this time differentiation between the two effects as well, which is nice. Another thing you can do with this technique is you can look at current source density profiles, and I haven't dug super deep into this yet. But just from the surface, you kind of you take this second spatial derivative, you can get an estimate of local current sinks and sources, and just from a kind of 35,000 foot level observation, you can tell that sort of there's definitely a different deviant response than there is to the control, and then also contextual redundance also have a significant decrease in their current source density profiles as well. So there's a lot to kind of dig into there, which is yet to be explored. And then also, sort of in the human literature, in the human EEG literature, one thing that's been identified in this paradigm is that by breaking things into the time frequency domain, you get a much more reliable estimate of context processing. So what this is just taking a single trace and using sort of moving window Morley wavelets, decompose the single trial LFP response into a time frequency plot. So here you have time on the x-axis, this is milliseconds, and you have frequency on the y-axis. And this is the typical response to one of these visual stimuli in the time frequency domain. So you had this nice kind of low frequency increase in power, and you also had this sort of gamma band increase in power too to a visual stimulus. Now what's interesting is when that stimulus is redundant, you don't see a whole lot of modulation in the low frequencies, but you see kind of a suppression of this gamma band response. Whereas when the stimulus is contextually deviant, most of the modulation is down here in the low frequency. So not only do we have a time, in the LFP, not only do we have sort of a time differentiation, but we also have a frequency domain differentiation. Deviance detection appears to occur mostly kind of in your theta, alpha, low beta oscillatory range, whereas adaptation tends to occur a little bit more in your gamma band range as well. So to summarize that sort of, oh yeah, so this is just the same data, but plot sort of averaged over time after the stimulus. Here you have frequency on the x-axis and power on the y-axis. So you can kind of see how the contextual deviant amplifies power kind of in your low frequencies, whereas your redundant stimulus results in a modulation of gamma band activity. So if we kind of just summarize this here, we know when the stimulus is contextually redundant, you get a suppression of responses in neuronal spiking, and you also get a suppression sort of gamma band activity. Whereas when a stimulus is contextually deviant, you get an amplification of responses in single neuron spiking and also a increase in low frequency activity. So to summarize that first part, can we study context processing in mice in local sensory cortex? I would say yes to some strong degree, and even to some degree looks a lot like the human mismatch response. So now that we have this model, now we can start to then look at individual levels and manipulate with dreads and optogenetics and different optical techniques. So the first part, I'll discuss how sort of context processing is carried out by local interneurons in the visual cortex. So the majority of neurons in neocortex are pyramidal cells. So these about 80 to 85%. And these are excitatory neurons, and they have relatively stereotyped kind of morphology and firing patterns, and they synapse both locally and distally. Whereas a subset of your neurons in neocortex are what you call interneurons, and these tend to be GABAergic, so they're inhibitory. And there's a large variety of subtypes. And the two I'll mainly be focusing on today are somatostatin-containing interneurons and parbumin-containing interneurons. So parbumin-containing interneurons make up about 40% of your GABAergic cells. And they kind of broadly target all cell types in the local area. They target both pyramidal cells, somatostatin interneurons, and even themselves, other PV interneurons, as well as they have authapses as well. So they're kind of broadly applying blanket inhibition. And they have a much faster firing rate, and they tend to support gamma band oscillations. Somatostatin-containing interneurons, especially within visual cortex in mice, tend to be disinhibitory. Whereas instead of targeting everybody relatively equally, their synapses onto parbumin interneurons are much stronger. So they tend to relatively selectively subserve disinhibition. So when they're active, they suppress PVs, which disinhibits local pyramidal cells. And they have a much more moderate firing rate, and they're thought to sort of support alpha and beta band oscillations as well. So what's particularly interesting about these two cell types in this paradigm is that both are known to be affected in schizophrenia. So looking at postmortem brain samples, we see, especially in visual and in auditory cortices, you see a significant reduction in PV mRNA and somatostatin mRNA, which brings up the idea that maybe sort of our context processing deficits might be due to deficits in the function of these subtypes of interneurons. So that was kind of the hypothesis. And so the goal was to first kind of suppress somatostatin interneurons and see how that affects kind of your mismatch negativity. So in order to do this, they used DREADS, which stands for designer receptors exclusively activated by designer drugs. And so what this involves is sort of virally targeting a sort of inert inhibitory receptor to somatostatin neurons, which typically doesn't do anything to the function of these cells. But then when you add an inert ligand, clozapine oxide, the combination of these two elements leads to the suppression of synaptic output from these cells. So we can sort of measure sort of context processing, deviance detection, and so forth first, and then apply the drug which suppresses somatostatin neurons and see how that affects the deviance detection. So here, this is before suppressing somatostatin neurons. Again, you have amplified responses to the contextually deviant stimulus in red, and you have suppressed responses to the contextually redundant stimulus in blue. And then after we suppress somatostatin neurons, on the whole, local pyramidal cells are still sort of responding to the stimulus, and you still have a stimulus-specific adaptation. You still have suppressed responses to redundant events, but you no longer have deviance detection. So taking somatostatin neurons out of the picture relatively selectively affects just this part of sensory context processing. That's what goes on at the single neuron level. When we then look at the LFP, again looking at time frequency responses, again, as I mentioned before, your deviance, so this is before suppressing somatostatin interneurons, and again, you have sort of frequency on the y-axis and time on the x, and these are responses to the stimulus in the neutral, redundant versus deviant case. And as I mentioned before, you have sort of amplified responses in the low frequency domain kind of track deviance detection, whereas modulation in the gamma domain track adaptation. So we have this frequency differentiation. This is the same data, again, just averaged over time. This low frequency augmentation is deviance detection, whereas stimulus-specific adaptation is this gamma band thing. When we suppress somatostatin interneurons, you still have stimulus-specific adaptation, just like you had in your single neuron responses, but the low frequency modulation is gone. This is just comparing the time average data back to back here. You still have this gamma band kind of suppression to redundant stimuli, both pre and post, but you no longer have this low frequency deviance detection response. To some degree, I've shown that aspects of deviance detection are dependent on the function of somatostatin interneurons, but what about PV interneurons, the other kind of cell type I chose to focus on? So use the same technique, use DREADS to suppress PV interneurons. And this is just focusing on basic sensory processing of moving gratings. And what you see, just to show that the suppression of PVs is working well, it disinhibits the early response. So again, if you have time on the X and frequency on the Y, it kind of disinhibits the early visually evoked response, but you no longer have this kind of sustained gamma band oscillation when you suppress PVs. So this is expected, and this tells us that our manipulation kind of was consistent with what the literature says it should do. Also, if we look at spontaneous activity after we suppress PVs, here the left and right show the same data, but the right is individual neurons. So these are non-PV cells. And this is the spontaneous activity before and then after suppressing PVs. So the identity line would mean that they have the same activity before and after. But that's not what you see. When you suppress PVs, as expected, you disinhibit spontaneous activity. So I show this to say, of course, the manipulation is doing something, but it's not doing anything to our context processing. So before and after suppressing PV interneurons, you still have augmented deviance detection, augmented responses to deviant stimuli, and you still have suppressed responses to redundant stimuli. So this deviance detection appears to be relatively selective, at least when we're comparing PVs to SOMs in visual cortex. So the question is why. We know that manipulating a schizophrenia-relevant cell type affects a schizophrenia-relevant biomarker. But this is sort of ongoing work that I hope to hammer out. We need to sort of record directly from each of these cell types during this paradigm. And sort of one hypothesis for why this might happen is, so we know that if you repetitively stimulate PV cells, what you actually get is an adapting response. So this is just sort of dual recording from a regular spiking cell to a fast spiking PV cell. And you see that sort of PVs adapt to repetitive stimulation, whereas somatostatin neurons do the opposite. So as you repetitively stimulate them, they get larger and larger postsynaptic potentials. So putting these two facts together with the fact that somatostatin neurons suppress PVs, you can imagine during an oddball paradigm, as you repetitively stimulate the local circuit, PVs are decreasing their activity, whereas somatostatin neurons are increasing their activity. And this would lead to kind of a general disinhibition, so that when the deviant stimulus comes in, everybody's disinhibited, and you get a sort of a super threshold response. So that's something to test, and that's a work in progress. So how is it carried out in sort of local circuits? Well, we know somatostatin neurons are involved. But they sort of zoom out and kind of, instead of focusing on the single neuron case, kind of get an understanding of how local interneurons interact during this paradigm. So again, you can see that we're able to simultaneously record a large number of neurons during this paradigm. And this is what it looks like if you just average over each one of these. But one thing we know is that there's a lot of variability from trial to trial in how individual neurons respond to the same stimulus. Even if two neurons had the same orientation selectivity, they're not necessarily going to fire exactly the same to the same stimulus across time. So also, if you look at this, you can sort of, or at least this was the thought process that we had, it's not exactly clear whether the same neurons are doing both of these modulations. Whereas on average, the whole population is kind of suppressing to redundant stimuli and amplifying to deviant stimuli. It may be the case that subsets of neurons do different aspects of this context processing. So in order to explore that, kind of looked at this variability and asked the question, are there subpopulations of cells or subclusters that kind of act differently across context and across time? So I can get into the exact methods I use to tease this out if someone's interested, but essentially you do k-means clustering analysis and ask the question, how well is the within cluster distances reduced if you add 2 clusters, 3 clusters, 4 clusters, 5 to the solution. And you compare that to a shuffled data set. And what this told us is that a solution with 3 clusters, or sort of 3 cell subsets, which do different things across these contexts, was kind of the most optimal. But we didn't really need this analysis to tell us that. If you just look at the 3D scatter plot, you can really see that there are 3 clusters there. Each one of these dots here is an individual neuron, and the x-axis is the average response kind of to the redundant stimulus. The y-axis is the average response to the control stimulus, and the z-axis is the average response to the deviant stimulus. So you can see that different subclusters of neurons seem to be preferring different contexts here. And so if we take that same data and we sort them out into their subgroups, that's exactly what you see. This first cluster here, we can call deviance detectors. And what the idea there is that they're mostly silent when a stimulus is redundant or kind of contextually neutral, but they really come online and fire only when it's an unexpected event. So we can call these deviance detectors, and they'll be in red. There's another subset that seem to be suppressed during the oddball paradigm in general, regardless of whether the stimulus is redundant or deviant. But they're typically active during kind of a more neutral context. So we can think of those as generalized adapting cells, or perhaps they're deviant suppressed cells. It's not exactly clear. And then there's another subset which doesn't seem to have any clear contextual modulation at all. If we just take this same data here and we just average within groups to simplify things, here are the 3 clusters. You have your general adapting cluster, which are relatively silent during an oddball paradigm, but they tend to be active during your typical multi-orientation paradigm. You have your deviance detectors, which are, I think, the most interesting subcluster. And then you have this kind of non-modulated or mixed modulation subcluster as well. And this wasn't just sort of an artifact of shifting fields of view or anything. So if you just take the same cells and you record again an hour later, the same cells prefer the same contexts. So your generalized adapting cells are still selectively active during the kind of multi-orientation paradigm, whereas your deviance detectors are still deviance detecting and so forth. So this was also true if you just take the even trials and then you make the clusters on that and compare to the odd trials. So this was pretty robust. And what's important is that sort of these cells are spatially intermixed. So if you just kind of plot where they are in the field of view, it's not like there are certain kind of subregions of primary visual cortex which have deviance detectors versus adapters and so forth. And again, it doesn't look like there's sort of a shift in a field of view. Your generalized adapting cells really are active during your control run, but they're pretty silent during the oddball run. And then vice versa, your deviance detectors, they're still in the field of view. Their signal's still there. It just kind of comes online during when there's a deviant. So another interesting aspect of this is that your subtypes, your kind of context-preferring cells, also show more correlation within type than between. So if you ask the question, kind of you have all these deviance detectors in the field of view, and you look at spontaneous activity when there's no visual stimulus, you see much stronger correlations between cells which prefer the same context than between cells which prefer different contexts. So it doesn't necessarily purely show that these cells are synaptically connected, and this is something I'd like to look at, but it at least suggests that these do form some sort of subnetworks, or context-preferring subnetworks. They could be sharing similar input, or they could be sort of parsing different interneurons, may target one type versus another. But this was relatively surprising because a lot of the ways we'd previously analyzed it, in the same way everybody else had previously analyzed it, sort of assumed that the cortex was being bidirectionally modulated instead of there being sort of subsets of cells that do different parts of it. So we also looked in auditory cortex. Here, again, using awake mice on a rotating treadmill, again using GCAM6S. But the difference is sort of the head plate has to be pretty far over on the side of the head, and which took sort of removing some muscle. But after we sort of worked those kinks out, it'll work very nicely. Another thing that you need to do when you're doing primary auditory cortex versus visual cortex is very large and easy to find stereotaxically. But with auditory cortex, you definitely need to do a mapping experiment first. To determine whether you're looking in primary versus secondary or tertiary areas. So you just do sort of kind of pure tone sinusoidally modulated gratings and map out your areas first with a 1-photon approach, and then you can focus, as we did, using 2-photon imaging just on primary auditory cortex. The question being, does primary auditory cortex and primary visual cortex both show the same sort of context-preferring ensembles? The stimuli we used for the oddball paradigm were not pure tones, but instead they were sort of these time frequency gratings. And just kind of on the surface, they look a lot like the visual stimuli that we used. And that was intentional because, so these stimuli go from 3 kilohertz all the way up to 90 kilohertz. And they're sort of sinusoidally modulated in time. I don't have an example right off the bat, but they do sound kind of funky. But the idea is that these stimulate the whole tonotopic auditory map. And so again, we can sort of use a flip-flop auditory oddball paradigm, and then also a many standards frequency control. So here is after averaging over all of your auditory neurons. Again, you have this bidirectional effect as you saw in visual cortex. These are calcium transients. You have your contextually redundant stimulus leads to suppressed responses relative to the context neutral control, whereas your contextually deviant stimulus is an augmented response. And then again, when we do a k-means clustering analysis, what you see very clearly is subsets of auditory cortical neurons prefer different contexts. So actually the largest chunk this is just from 3 mice, this is an ongoing work, but the largest chunk of cells actually were deviance detectors in primary auditory cortex. I'm not sure yet if that's going to hold, but if we just kind of compare back to back here, these were the subclusters in visual cortex, and these were the subclusters in auditory cortex, and you can see very nicely you have these deviance detectors in both cortices. You also have these non-modulated cells in both cortices. What's different is you don't have generalized adaptation in auditory cortex, and I'm not quite sure why. You have a much more specific thing. So this subset of cells isn't doing deviance detection. They don't amplify their responses to deviant stimuli, but they do suppress their responses to redundant stimuli. So this tells us both stimulus-specific adaptation and deviance detection can be studied in auditory cortex, but again, just like visual, this is sort of carried out by different subsets of layer 2-3 neurons. So how is sort of context processing carried out in local ensembles, or local populations? Well, it's done by sub-ensembles actually, rather than all equally in all neurons. Again, as was brought up, this has mainly been done in layer 2-3. So the next question is, what about if we sort of extend to layer 4 and layer 5? Do we still see deviance detection? Do we still see these context-preferring ensembles? Just kind of to summarize, your canonical neocortical circuit And this is highly simplified, and I'm leaving out layer 1 and layer 6 here. But the majority of your bottom-up input comes from, especially in primary visual cortex, comes from the LGN, the basic relay thalamus, to layer 4 cells, where it's fed forward to layer 2-3 neurons. And then sort of you have a lot of sort of local and lateral connections. You can think of this as this sort of local computation. And then these sort of synapse on layer 5 cells, which send driving input to higher thalamus, and also sort of modulatory feedback back to your relay thalamus. But sort of you can split this into sort of input, local computation, and output if you want to use such a heuristic. All the work I've done showed you so far is from layer 2-3, but working with Weijian Yang, who was a postdoc in the USTA lab, and now he's assistant professor at UC Davis. He sort of developed a technique where we could measure 10 different planes at the same time with 10 hertz imaging. So the rest of what I've showed you is with 30 hertz, but 10 hertz still gives us a pretty good picture of how neurons are responding to these stimuli. This involves injecting kind of a red calcium indicator in these deep layers and a green calcium indicator in the superficial layers, and then using 2 different lasers, one targeting deep to hit your JR gecko, and then one targeting shallow. And then he used spatial light modulators to sort of split this beam into different Z foci, and also electrotunable lenses to very rapidly switch the Z focus over time. So what this gave us is exactly that. A very large number of neurons from a nice sampling of cortical depth. And then sort of the goal was to record the same paradigm across these layers. So first off, this is from layer 2-3 again, and this in a totally different setup in somebody else's hands replicated the effect of sort of deviance detection is mostly carried out in a subset of cells, and they make up about 40%. So that was nice to see. And also you have your generalized adapters are also present in layer 2-3. So this was right down to the proportions of cell types was replicated what I saw in the previous experiments. But when we looked at deeper layers, you saw much less deviance detection. So on average, the number of cells which are classified as deviance detectors in layer 4 and layer 5 was about half to a third. And then if you sort of average over all the cells, there's really no deviance detection going on in these deeper layers, which was kind of interesting. It seems like it's mostly a layer 2-3 function. So here, the majority of cells are kind of active in your control paradigm and then largely suppressed during the oddball paradigm. So exactly why isn't clear, but this is just kind of another depiction of what was going on. Here, the x-axis is depth. You have layer 2, 3, 4, and 5. And these lines indicate the absolute number of neurons that are present from each cluster in each one of these layers. So you see the generalized adapter is in gray, and they're relatively the same overall number across areas, but you have much fewer deviance detectors. This dotted line just tells you the relative, so I'm missing another z-axis here, but tells you the relative proportion of cells in each layer which were driven by the visual stimulus. So that wasn't really different. What was mainly different was just the absolute, the relative number of deviance detectors. So deviance detection on average, and also the proportion of deviance detectors, is much more of a layer 2-3 phenomenon. So that's something to kind of delve a little deeper into as well. Okay, so we have kind of what's going on, what sort of contribution interneurons play, what's going on when you consider populations, and what's going on when you consider different layers of cortex. And sort of the next scale to look at is when we consider different brain regions. So visual cortex is not an island, but it receives a lot of sort of bottom-up input from the thalamus and also top-down input from prefrontal areas. So this was the next level of function to kind of depict context processing at. So what I mean by bottom-up is I mean sort of inputs directly from DLGN thalamus. And this is a relay thalamus, so they receive their primary input from the retina and the eye. So this is kind of the initial relay of sensory information to visual cortex. So the question is, what are these inputs doing during this paradigm? Are they carrying deviance detection or not? And then also asking sort of our top-down circuits. So we know mouse prefrontal area CG, or ACA, which stands for anterior cingulate area in the mouse. This sort of higher level cortex projects directly to mouse V1 and to layer 1 of V1. So we asked the question of how are these two subcircuits contributing to context processing? Do they kind of contain these sub-ensembles or not? Or are they causally related? So how do we probe these inputs? Well, one way to do it would be to sort of express your calcium indicator GCamp and then carve out cortex and focus your microscope directly on LGN. But you damage a lot of tissue in the process. But another way to do it is just sort of express your calcium indicator and then focus the microscope still on V1, but making sure that you're very kind of careful about leakage and spread. You can actually just kind of directly observe these boutons and directly observe the sort of axons from DLGN to V1. And so this is kind of some real-time data. This is in visual cortex, but these are thalamocortical axons. And you can see you can resolve individual axonal branches and boutons. So this is just spontaneous activity, and it's nice and clear. The signal-to-noise ratio by using this approach is comparable to that using by using a calcium indicator and focusing on the soma. So here's sort of your DF over F trace of sort of burst firing in your V1 neurons, and you have the same thing in these sort of axonal inputs. So using this technique, we can actually get information of about what signals are being sent directly from your relay thalamus to visual cortex. And this is what we did. So again, using the same paradigm, here is responses of the same sort of thalamic axons to the same stimulus when it's contextually neutral versus redundant versus contextually deviant. And so you have, right off the bat, you can see that the kind of modulation isn't quite as dramatic as what we saw on V1. When you average over these thalamocortical inputs, you definitely see stimulus-specific adaptation. So the inputs from thalamus to V1 are still suppressed when the stimulus is repetitive, but you no longer have deviance detection in these inputs. So the red line is the average response to the deviant stimulus is right on top of the context-neutral control in these thalamocortical inputs. If you just kind of compare this back to back, this is what was going on in the V1 neurons. You had amplified input, or amplified responses to a deviant stimulus and suppressed to redundant. You only have that redundant suppression in the thalamocortical inputs. So no deviance detection at the population level when you average over inputs to V1. And then if you do the same sort of k-means clustering analysis, this was the result from your V1 neurons. Your thalamocortical inputs, you only kind of see two clusters there using the same approach. And those two clusters, neither one of them is doing deviance detection as well. So you have this kind of smaller subset which doesn't modulate with regard to context, which we're seeing everywhere. But the rest of them, about 2/3, were just your stimulus-specific adapting. So neither one of these subsets was doing deviance detection. So this tells us some aspect of, this tells us that deviance detection isn't kind of very simply and directly inherited from the thalamus. It's at least computed after the thalamus, somewhere in visual cortex or beyond. Yeah?
Are you still using an orientation stimulus?
Yes, yeah.
LGN's not orientation to it, right?
It's orientation to, yeah, right. So, well, actually, there is some orientation tuning in these cells.
It's not as strong as it.
It's not as strong, yeah. So, right. At the very least, we don't see deviance detection therein. But I mean, it just at least tells us it's not simply inherited from there. But yeah, that's right. In the mouse, it's a little more than higher mammals for sure. But yeah. So there may be some other stimuli that would be nice to test and see if they're also showing. Yeah, they're also showing this, right? But I did see some orientation tuning among these inputs. It definitely wasn't as robust for sure. Okay, so So yeah, just to summarize that, we see not only deviance detection and adaptation in layer 2-3, but these nice kind of segregated sub-ensembles preferring different contexts, whereas in your bottom-up input from the thalamus, we have neither deviance detection nor deviant preferring ensembles. So that's the bottom-up story. What about your top-down inputs from PFC? Again, we use the same approach. So you express GCamp only in these cells, and then you focus your recordings on layer 1 of visual cortex. And these are, again, sort of axonal segments and boutons from PFC neurons in visual cortex. So doing the same analysis here, each row here is an individual anterior cingulate axon, which is sent back to V1. And we're using oriented stimuli again. And you see right off the bat, you see definitely clear visually evoked responses in these top-down inputs. And you see what appears to be, at least in a subset of them, stimulus-specific adaptation. So these top-down inputs do have decreased responses to redundant stimuli, but again, they're not on average doing deviance detection. It doesn't appear so. So that was somewhat surprising because we suppose that perhaps this deviant signal is computed later on and then sent back to V1, especially given how late it occurs. But it doesn't appear that, at least once you average over all these PFC inputs to V1, that they're doing deviance detection. And then this is just, again, directly comparing the PFC responses to V1. You don't have that amplified response to deviant stimuli. Now, when we did the k-means analysis, the story gets maybe a little bit more complex. So again, you have these non-modulated inputs from PFC to V1. But you also have this subset, which is doing kind of a bidirectional modulation. So it's almost as though these cells are kind of relaying back exactly what the average V1 response is back to V1. They're doing both deviance detection a little bit and stimulus-specific adaptation. So there is this bidirectional subcluster in PFC, which leads us to believe it could be at least to some degree contributing to what's going on in V1 during this paradigm. So that's where the story sets right now. We know that sort of somatostatin interneurons play a role. We know that this is not, this is sort of different contexts are preferred by different subsets of cells. Deviance detectors are more common in layer 2-3. And this is directly inherited from DLG and thalamus. But the kind of the remaining question that's one which is mainly unanswered by just observing the activity is sort of, does the top-down PFC inputs play a causal role either in your average responses to different contexts and also your context preferring ensembles? So in order to test this, use an optogenetic approach. Again, this is recording neurons in V1 with calcium imaging, but ARCH-T was expressed in PFC neurons, and then kind of recorded our oddball paradigm in V1 neurons, and then had another run where we suppressed the inputs from PFC to V1 using optogenetics on every other trial. So this is what looks like a baseline. I've shown a lot of plots like this. This is, again, you have your increased responses to deviant stimuli. These are V1 neurons, and suppressed responses to redundant stimuli, as expected. When we suppress PFC inputs to V1, we lose deviance detection. So there's no longer amplified responses to a deviant stimulus. You still have stimulus-specific adaptation. If anything, it appears a little stronger. But your responses in the neutral case are essentially equal to your responses in the deviant case. Almost as though now all stimuli, if they're rare, they're unexpected. Or so they're treated as deviant. This also makes your V1 neurons look a lot like the average input from thalamus, which is interesting. And this could be due to two different effects. You have suppressed responses now. When you take PFC out, responses to deviant stimuli are smaller, but also responses to control or neutral stimuli are larger. And this is the same data just with bar plots here. At baseline, you have deviance detection and adaptation, whereas when you suppress PFC, you only have your adaptation, but you no longer have deviance detection. So that's when you average over all neurons. But suppressing PFC didn't really affect kind of ensemble membership, which was interesting to us. So you still had the same relative proportions of deviance detectors and the same relative proportion of adapters before and after taking PFC inputs out of the picture. This is just kind of the same data, but you can just kind of directly see this is before suppressing PFC inputs to V1. You have your deviance detectors and your adapters and your non-modulated. And this is after suppressing PFC. You still have those cells still retain their same identities. Your kind of context preferring subensembles in V1 still prefer their preferred context. The main thing that's changed is their relative response magnitudes. So this is before and after suppressing PFC. Deviance detectors seem to decrease their responses, whereas your generalized adapting cells tend to increase their responses. So kind of flipping this, you imagine PFC seems to be suppressing these cells, whereas it's sort of exciting these cells, either directly or somehow indirectly via disinhibition. And so that needs to be worked out as well. How can you go from kind of a bimodulated subcluster and a non-modulated cluster to these nice kind of relatively complex effects on these subclusters here? And the sort of non-modulated cluster wasn't really affected much. It was slightly decreased after you took PFC out of the picture. So this needs to be worked out kind of, and I think a nice way to do this would be first by modeling and getting an idea of what could be going on. We know a lot about who somatostatin interneurons target, also subsets of somatostatin neurons, who they target, and where and when. I haven't looked at or explored VIP interneurons, but they could be an interesting candidate, as well as PVs. The first sort of work out how this circuit could underlie this complex effect. And then sort of stimulate PFC inputs while using DREADS to suppress different subtypes and seeing how that affects it. So there's a lot of experiments that kind of explore this model and get a deeper understanding. And it also would be important to kind of measure time frequency responses during this paradigm as well, to link not only what's going on at this kind of single cell and multi-cell level, but also match it up to something we can measure in patients, sort of oscillatory responses to these stimuli, to kind of get a nice full picture of how specific things affecting individual interneurons might give rise to what you can observe with a non-invasive EEG. So that's kind of the bigger picture here, but instead of focusing on a mouse model of a very complex disease, we can sort of focus on a particular domain of dysfunction, which we can translate to mice, and start to kind of link up how individual cell types and circuits and perhaps sort of neuromodulators give rise and modulate this response to get a bigger picture of that function rather than a complex disease. So that's everything. So I need to thank my postdoc advisor, Raphael Juste. Yuri Shimkiv was a PhD candidate who worked with me on a lot of this, especially the auditory stuff and the thalamocortical inputs. And also sort of Wei Jian and Shuting Han, they did a lot of the technical and actual experiments in the multiplane imaging in this paradigm. So thanks, everybody. Oh, that was for some other reason. Yeah, that was a good catch. I should update that. That was one way I originally had conceptualized this, that somatostatin neurons might disinhibit the circuit both by suppressing PVs and VIPs. We know SOMs and VIPs have this kind of mutually antagonistic relationship. And so that was one way I started to think about it, just to simplify the picture. but yeah, no. In this case, actually, in no line I'm aware of are they targeted. PVs and VIPs are targeted, and not SOMs. It's yeah, yeah.
After you induce this chemically induced depression of the deviation, were you able to see any behavioral changes in these animals, or was it a transient?
Behavioral changes due to the somatostatin suppression? Oh, that's a good question. They didn't change sort of locomotion, and that was more or less the only thing we quantified. There wasn't any sort of very obvious changes in their behavior. And again, it's very selectively suppressing somatostatin interneurons in visual cortex. And so it's, it is possible that they might behaviorally respond to visual stimuli different, but their overall behavior wasn't changed. And it is also true that some of CNO is converted to clozapine, which is an antipsychotic. The thought being that it's not enough to affect their behavior. And if you do the right controls, you can really kind of tease out what are the effects of CNO/clozapine on some interneurons versus the generalized effects. But yeah.
So it's interesting that the deviation detecting neurons don't really remap with the PFC inactivation, right? And I guess on some level, there's still a really strong deviation response. It's not different anymore from the adaptation effect. So how does the computation play out then? Is it something like this at the broader level, the circuit is actually comparing the relative magnitude of general adaptation and deviation effects, and that's sort of the evidence that the deviations occur?
Yeah, that's a good question. I mean, I don't know. It would be nice to see sort of on a downstream target whether deviance detectors and your more general cells synapse on the same cells, or do they actually continue to form different subnetworks? I'm not sure. So so this particular paradigm has in the past been used to say, patients showed disruptive mismatch negativity, and this is potentially due to PFC abnormalities. When you're looking at the aggregate, yes, and if you just sort of average the overall V1 output, it does look like PFC is doing that. But there's still some computation that's ingrained in the circuit. The circuit is still detecting deviant versus non-deviant. So I think there could be many ways to lead to abnormal context processing at the behavioral level. But yeah, it would be good to know. And I would suspect they probably do target the same neurons. And there is kind of a weighting that happens, but I don't know. Say it again? So using, is the question about using transgenic animals?
Other animals.
Oh, other animals. Oh, like rats and primates and. Well, so the mismatch negativity has definitely been studied using kind of EEG in rats a lot, but it hasn't been done. They haven't looked at specific cell types or circuit manipulations at all. Usually that's done in a very clinical setting, sort of not clinical, but very much we'll just record one trace and we'll just compare the redundant to the deviant and see if there's a difference. So I think there's a lot of really good work that needs to be done in other animals as tools are developed, even as basic as using kind of more thorough experimental paradigms. Or as measuring different layers of cortex. And definitely not in non-human primates has this kind of been done in the right depth, I think. So.
I'm going to ask the last question.
Yeah.
I was wondering if you've examined how your 3 clusters of neurons are related to their orientation to.
Oh, yeah.
So is it as simple as some are sharply tuned, some are broadly tuned, and that's how they cluster?
Yeah, so that's a good question. So I did. Your non-modulated cluster is, they tend to not be orientation tuned at all. So they're more or less responding to all. So they're neither context tuned nor orientation tuned. but both of these subsets do show orientation tuning. The one issue is in order to assess orientation tuning in this paradigm, you need to do many, many paradigms, because you need to have a paradigm where this stimulus is the deviant, and then where this stimulus is the deviant, and where this one is. But as far as I can tell, they're still showing about the same orientation tuning as your generalized adapters. So not only are there deviance detectors, but there are deviance detectors for specific stimuli, which is even more interesting. There's sort of nested subnetworks there of different preferences.